Welcome back to the course Fundamentals of General Immunology. Today we'll discuss a few of the interesting facts about the topics which we already covered during our course, and we'll try to see some of the important facts about the reactions or some unique thing about the immune system which was not covered in the routine lecture. So let's try to move on and see some important facts about immunology. So what we understood is a presentation of an antigen. When we are saying anything in terms of antigen presentation, there are other MHC1 or class 2 MHC molecules are associated with it. And what generally is known that when you have an antigen, suppose this is a cell, and if there is an antigen which is outside, we call it exogenous antigen. And when if it is exogenous antigen, generally it enter inside the cell and process through this MHC2 molecule. While if the antigens are prepared internally, we call it endogenous antigen. So these are endogenous antigen. And if it is endogenous, then these are presented through MHC1 pathway. So this is what we have understood during the course of our antigen presentation, and both these antigens are different. Most of the time when we talk about endogenous antigen, we are talking about viral specific antigens, while when we discuss anything related to exogenous antigen, we mostly talk about bacterial antigens. So this is bacterial in origin. Now in certain conditions, we may see that there could be a cross presentation where the exogenous antigen can be presented through class 1. So what we are trying to understand here is a phenomena called cross presentation of an antigen, where MHC1 specific molecule can present the antigen instead of endogenous antigen. They can start presenting the exogenous antigen. So that is what is called cross presentation, and these are not done in all of the cell type because most of the cells are expressing MHC1 molecules, and we expect them to process through MHC1 pathway, but only two distinct cell population in the immune system, particularly the dendritic cell. So one is called plasmacytoid dendritic cell, the other is called conventional dendritic cell. These two cell populations are only cross present the antigens. We will see in a while how this phenomena can happen. While the plasmacytoid dendritic cell are basically the cells which are generated upon the virus infection. They are well known to produce type 1 interferons. So type 1 interferons are either alpha or beta. So this is typically producing most of those type 1 interferons, and they are generated in response to the viral infection in the human body. While the conventional dendritic cell, mostly what we understood as a dendritic cell, are mostly antigen presenting cell, and they can present the antigen to the immune cell in order to raise an immune response. So, but in certain cases, these dendritic cells can cross present the antigen. I mean, the exogenous antigen can be processed through MHC1 pathway, as what we discussed just now. So we'll see how this happens, what are the molecular mechanism behind this cross presentation, and why this is important. So let's try to see what are the different types of dendritic cells available in the human body, and in mice also. So let's try to see the dendritic cell classification. So there are many sets of dendritic cells which are available in the body. So one is called plasmacytoid dendritic cell, which we just now discussed. Conventional dendritic cells are of two types, type 1 and type 2, based on the markers which they express on the cell surface. Monocyte derived dendritic cells are the other type of dendritic cell, and then Langerhans cells are also type of dendritic cell which are present in the skin area. So these are all phagocytic cells, because if you see the morphology of these cells are having prolonged cytoplasmic processes which can allow the antigen to present and particularly do a phagocytosis process in order to see what kind of antigens are entering. While in case of plasmacytoid dendritic cell, we see a very plain morphology, no dendrites. So these are behaving like a plasma cell, and they can generate cytokine essentially to clear the infection. And mostly they are activated upon viral infection. They are known to secrete type 1 interferon mostly, but sometimes they can also secrete type 3 interferons. So we can see type 1 and type 3 interferons they can produce. While conventional dendritic cells are mostly antigen presenting cells, so they can present the antigen to the immune cell and can induce cell mediated or antibody mediated immune response. So these are actually against extracellular or intracellular antigens which are present in the nearby area. While if we see these cells, how we can differentiate these cell types, although they look all similar, but they are differentiated based on the proteins which are expressed on their surface, and therefore we generally called it cell markers. And each cell type, you can see there are many cell type specific proteins which are secreted, all present over the surface of these cells, and they are quite divergent, and based on that we can classify this dendritic cell. But our focus is mostly on these two cell types which are involved in cross presentation. But just for the knowledge, we can understand that these are some of the conventional or dendritic cell types which are present in the body. In terms of their origin, they are mostly say plasmacytoid dendritic cell, conventional dendritic cell, and the basically both the types of conventional dendritic cell are produced in the bone marrow. So we see bone marrow is the actual site of origin, while monocyte, since its name itself indicates, these are derived from monocytes, so monocyte can be converted into a dendritic cell. So obviously their location is mostly on the blood system, while the Langerhans cells are mostly present in the tissue level. So they are actually the phagocytic cells of the skin or any other area. So we can see that in the fetal liver and yolk sac also these are expressed in a large quantity. While their location wise is the same, they can express in the lymphoid organs and the blood system. However, the Langerhans type can be present in the epidermis or the mucosal epithelium. So these are all different types of dendritic cell. They are known to process the antigen. They are antigen presenting cell. Some of them are also involved in cross presentation, and that is what we are discussing here. Let's try to see and understand the molecular mechanism of this cross presentation. So when we call cross presentation, there are two pathways. One is a vacuolar pathway and the other is cytosolic pathway. The vacuolar pathway is the one where the phagocytosed antigen can go to the vacuoles. So what's happening that these are exogenous antigen, one can see it coming from outside, so this is exogenous, and once these exogenous antigens are coming in contact with the plasma membrane, these phagosomes are formed and they can engulf these antigens into it. And once they are engulfed, they are processed into small peptides which are loaded over the MHC1 and can directly present on the cell surface in order to activate the CD8 specific immune response, because MHC1 is basically targeting CD8 specific immune response. Now this is a straightway vacuolar pathway where the antigen can be processed to vacuoles, and what we are trying to say, it's a cross presentation. And what is cross presentation is basically this should process through MSC2, but these are not processed through MSC2, but these are processed through MSC1 because of the cross presentation phenomena. And this cell we are talking about, dendritic cell population. Now the second pathway is cytosolic pathway, which is basically involving the cytosol, and cytosol is actually the part of the cell apart from nucleus which are there in the cell. So we can just try to understand that once the phagocytosis can happen, so we can start from here. So this phagocytosis can engulf the antigen and it can go to the phagosomes in the same way, and in the phagosomes these all smaller antigens can be formed because this is what is actually processed, or else they can pass through this proteasomal degradation pathway where the smaller peptides can be made, and this peptide can be transported to the transporter of antigen processing, TAP protein we used to call, and once it is transported into the phagosome, they can attach towards the MSC1 molecule and then drag to the cell surface. So this is one way. So once the cytosolic pathway is activated, the endogenized antigen, which is coming from the exogenous origin, once they are entering, they are processed through the proteasomal degradation, and once the peptide is made, these peptides are loaded over the MSC1 molecule while they pass through the TAP protein, which is actually a transporter of the peptide into the lumen of the phagosome. Or else there could be a possibility that these all peptides can process through endoplasmic reticulum lumen, where we have the MSC1 molecules already present, and once these proteins degraded by the proteasomal degraded smaller peptide can be loaded, pass through the lumen and then loaded on the MSC1 molecule, they can drag itself to the surface in order to show it to the CD8 positive T cell population. So this is the second way they can do. So either they can go through direct phagosomes activation, or they can pass through the endoplasmic reticulum and do like that. There could be other possibility where endoplasmic reticulum and Golgi intermediate complex, which are very important trafficking molecules in the cellular system. So some of the TAP can be transported from ERGIC complex, and with the help of this syntaxine 4 or Sec22B, which are actually the snare protein which are involved in the intracellular vesicular docking and membrane fusion along with the trafficking, can help these TAP protein to go to the phagosome. And therefore, we see that the whole phenomena can happen where this TAP protein can help to give passage to the peptide onto the phagosomes. So essentially, you can see that there could be a dual phenomena that can happen. Either the peptides can be presented through the vascular pathway, or they can present through the cytosolic pathway. And in both the cases, MSC1 will be activated, although these are exogenous peptide antigens. And once the antigens are processed in a smaller peptide, this peptide can be loaded into MSC1 either in the ER complex or through the ERGI-C complex dragged to the phagosome, or can be directly loaded over the phagosome directly. So there could be multiple ways this antigen can be processed in the dendritic cell, and this is very important because we generally understand that once there is an exogenous antigen coming into the system, it can raise a CD4 specific immune response, which means that they can generate a lot of antibodies. But once there is a cross presentation, we can see that the cell mediated immune response is also raised against these kind of pathogens. So that means this is a host mechanism where both antibody and cell mediated immune response can be generated against the same kind of antigen. So this is an important phenomena that can happen only in the dendritic cell, not in all cell population, although each and every cells of the human body which is nucleated will express MHC1 molecules. So we'll move on and see some more interesting facts about immunology. And one such thing is called ghost cell. This is basically a cell, particularly an RBC without, so this is an RBC without hemoglobin, So this is actually a white RBC, an RBC without red color, because somehow the hemoglobin is removed from this cell type. This is very important cell types which are generated in research lab in order to study the membrane biology, because RBC is a very good tool to study the membrane integrity, the protein, integral proteins and all. And by removing the hemoglobin content, this RBC can be studied for its membrane integrity in terms of protein and all. So there could be a lipid bilayer. These are phospholipid bilayer which can provide the structural framework to the RBC. Of course, it is the biconcave structure. There could be a lot of proteins which are present on the surface, peripheral proteins. There could be an actin and the spectrin network, the cytoskeleton protein, which could be present in the RBC. There might be a lot of carbohydrate surface glycoprotein chains which can help in the recognition of different kind of blood cell types, such as ABO antigens, which are known to differentiate between the different types of the blood group. There could be an integral protein which can pass from the whole membrane, and they can act as a channel protein or some kind of a important function they can do. So these ghost cells are important in terms of understanding the membrane trafficking as well, and see that whether anything is perturbing the cell membrane. Do we have any molecule that can pass into the cell membrane? And we can see doing some of the unique studies of the fluorescent biology, because these are basically colorless. So now we can generate some color and see what is happening. Now there are ways to generate the ghost cell by certain techniques, and since this is mostly available in the research, people are trying to generate this ghost cell by just changing the osmolarity of the media where the RBC can be kept. So generally we can collect the blood. So the first step to make the ghost cell is by generating kind of an EDTA containing blood, so that the blood won't clot. So we can collect the blood using EDTA or heparin, which is an anticoagulant, and once we can spin it, we can generally see that there could be an upper plasma layer and the buffy coat layer, which is WBC, and RBC will be there. Essentially we can segregate the RBC, and then we can hemolyze the RBC. So RBC can be ruptured by using some of the hypotonic solution such as phosphate, 5 millimolar phosphate buffer saline at pH 7.4. So once we keep the RBC into those buffers, the buffer will make this osmotic disturbances in the cell membrane, and then the cell will lyse and the hemoglobin will be out, and then we can wash it for a while after incubating it with the hypotonic solution, and then we can remove the cellular debris and the hemoglobin content, and then we can get the pellet here. These pellet can be washed and can be used as an ghost RBC, because the RBC membrane once ruptured, since there is a fluid mosaic model of the membrane, they can immediately join to and sealed in order to make a spherical structure again. So this is a very routine method people are using in order to remove the RBC, because of the general content based biology where the hypotonic solution is allowed to rupture the cell and then the content will be out. The whole cell which are devoid of hemoglobin can be tested for some other important research questions. Now these are some of the things which are unique in immunology, and one such thing is ghost cell. Let's move on and see something else, and one such important term is routinely will come eventually in this course is called precipitation, and the other is called agglutination. So we'll try to understand the difference between these two terms, precipitation and agglutination. So precipitation is especially a term which is used for soluble antigens. Remember, we are trying to understand these two terms in context of immunology. Precipitation is a term which is used for soluble antigen. So we can mark here the term soluble, while here it is a particulate antigen. So when we use a soluble antigen, it forms an insoluble precipitate with a specific antibody. So that means once we have a soluble antigen, and if we add the specific antibody that can bind to this antigen antibody reaction can form a precipitate which could be insoluble. While in this case the antigens are in the particulate form, such as bacteria or RBC and all, and this again can bind with a specific antibody to form this kind of an agglutinating structure. So there are differences in terms of antigen used to make this process. One is soluble, the other is particulate form. Next is, so what is happening in terms of its reaction altogether is, once we add, we can see that before reaction it is a kind of a clear solution, and after reaction we can see that the small precipitate can form in the bottom, and due to precipitation it can just settle at the bottom. While in case of agglutination reaction, we can simply see that there are lump of small masses of the things which are just settled at the bottom. So these are actually done in order to do some of the antigen antibody reaction in very routine condition that people are using in many routine processes. So something called ring test, which is actually an antibody reaction in a test tube. There is something called gel diffusion assay, and this is also called as Octolini test, which is also used to see the specificity of an antigen binding with an antibody. And also there could be other tests also, such as flocculation test, in order to check the antigen antibody reaction. Remember, these are all types of precipitation reaction because the antigen here used are all soluble in nature. While here we see that the antigens are actually particulate in nature, and we do see these reactions when we do a blood typing, such as ABO, Rh positive, negative blood typing. Again, Vidal test, which we will see in a while, which is used to see the typhoid positive cases in a patient. So these are very specifically agglutination reaction done by using the antibodies. These are very specifically reaction that is used to check the antibody against Salmonella and other bacterial antigens also. Latex agglutination test is another type of the agglutination test, which is generally used in order to see if there is an antigen antibody reaction, specifically the agglutination reaction. So we can just summarize the whole process as, in case of the precipitation reaction, the antigens are actually soluble in nature, while in case of agglutination it is mostly particulate, such as bacteria, latex beads or RBC, while antibody here is mostly bivalent or multivalent in case of precipitation, while in case of agglutination we see generally IgM or IgG can do this phenomena. Reaction visibility, of course, it's in the form of a pellet or a ring, while in this case we generally see that the visible change can happen because of the granular formation and all. Some of the example which we also always discuss, that we have Octolini test or the precipitin test, and here we have a Vidal test. Let's try to see what is Vidal test and how the agglutination reaction can tell us that the person is positive for the typhoid or not. So in the Vidal test, we generally use the serum from a patient showing some symptoms of typhoid. So if we want to diagnose a patient whether he is positive or she is positive for the typhoid or not, we simply collect the serum, and if you recall back, serum is collected from a clotted blood. So once we clot the blood, we can collect the serum from the top of that clotted blood mass. Then we can add two of the antigen coming from Salmonella typhimurium, and one is called O antigen, the other is called H antigen. O is actually somatic antigen, the H is called flagella antigen. So basically O is coming from the outer membrane of Salmonella, while H is coming from the flagella. So flagella is an organ in bacteria that helps in the motility of that bacteria. Bacteria will be motile because of the presence of flagella. And then we can set up a reaction where we can just use those antigen, O antigen and H antigen, and we can add some of the serum of the patient on top of it. And then we wait for a while and see what is the visible changes we are getting on this mixture. So if we see that there is a reaction, that means if the person is positive, the person body might have an antibody against either O or H antigen, because suppose if it is a bacteria, this bacteria will have an outer surface protein which are present on the surface, or will have a flagella that helps in the motility of that bacteria. So this antigen or this antigen, if it is present in the body will respond to this by producing an antibody. And if these antibodies are present in the human body, we can get it in the plasma. And we visibly see that if there is a change in the process after adding this, we can get agglutination in O and H, while in the negative case we simply see the antigen just lying plain. We won't see any agglutination to happen there. That means this is a very straight way, the visible eye specific test where agglutination reaction can tell us that person is positive or negative for a reaction. Now this kind of reaction can happen in most of the immunological reaction that takes place in vitro condition. So we can just collect the serum, we can add the particulate form of the antigen, there is a precipitation reaction, we can understand, or even in the soluble form of the antigen, if we just see the test ring formation or any kind of precipitation reaction or say agglutination reaction, we can say that the people or the person might be suffering from the disease. Although this is not a 100% proof of the infectivity, but most of the time this is positive in terms of antigen antibody reactions. So these all are very unique facts about the immune system. So what we studied today is the way antigen, the exogenous antigen, can be presented by a dendritic cell, which we call cross presentation. Generally, we won't see a cross presentation to happen with an exogenous antigen in normal cell, but there could be a possibility that exogenous antigen may be processed through MSC1 to activate CD8 specific immune response, while there could be a possibility that the CD4 specific immune response will always be there. So just to mount a cell mediated immune response, the body started getting this kind of unique process of cross presentation. Second, we discussed about the ghost cell, which is actually a hemoglobin less RBC. That's basically the outer covering of an RBC without hemoglobin. People are using that ghost cell in order to trap the specific dyes or look into the membrane permeability of a protein to see what's happening, what is the life cycle of a protein, whether the protein is a surface protein or internal protein. People are just looking into membrane biology using those ghost cells. And then finally we looked into two specific terms which we'll use very often in discourse, called precipitation and agglutination. And as I told earlier also, very specifically immunological precipitation and agglutination reaction. The only basic difference between these two processes is type and quality of antigen. If it is soluble antigen, we call it precipitation. If it is a particulate, we call it agglutination. And there are a number of different tests. People are using those two assays in order to identify a disease condition. And one such example we discussed here is to detect typhoid, and that's called Vidal test, where we can just detect the O or H antigen coming from the bacterial surface. So this is all about some of the facts which are there. We'll try to discuss some more interesting facts as and when we go ahead in this course. Thank you.